Це наші зустрічі, лекції проходять в рамках такої програми Малої академії наук «Лекції майбутнього». Раз на 3 місяці ми запрошуємо Нобелівського лауреата, і він зустрічається з молоддю, з школярами, студентами. І це вже 3-я лекція в Київському університеті. Я думаю, був хтось на попередній лекції професора Оффена? О, були. А хто це студенти всі, так?
Студенти, викладачі тут.
Так. Хто мав відношення до Малої академії наук? Чи ходив на конкурси? О, бачите? Так що це ваш захід. Мала академія наук для вас організовує ці лекції. Ви скористайтесь цією можливістю. Професор сер Пол Нерс, лауреат Нобелівської премії з біології і медицини. Нам його представлять, але це дуже видатна постать у науковому світі. Він був президентом Королівського товариства, член багатьох, зарубіжний член багатьох світових академій, почесний президент Рокфеллерівського інституту, якщо хтось чув такий. Хтось чув про такий Рокфеллерівський інститут? Не чули? Це дуже...
Лекцію нам зробити буде про світові...
Так, то подивіться в інтернеті, це дуже видатна наукова установа, подивіться в інтернеті. Ну і зараз професор... Після лекції у нас буде дискусія, ви зможете задати запитання, тому підготуйтесь, щоб задати такі запитання, за які не буде соромно нам перед професором. Так, представ.
З задоволенням хочу запросити до слова випускника Київського національного університету імені Тараса Шевченка, більше того, аспіранта Київського національного університету імені Тараса Шевченка, а за сумісництвом керівника Асоціації випускників Малої академії наук Євгена Кудрявця, який представить нашого гостя.
Доброго дня. Я всіх вас вітаю на черговій, це вже 13-та лекція майбутнього і 3-я лекція Нобелівського лауреата в рамках цієї програми. В цей раз в нас наш гість із Великої Британії, це сер Пол Нерс, який в 2001 році отримав Нобелівську премію в області медицини і фізіології. Крім того, сер Пол є 9-тим президентом Рокфеллерівського інституту університету в Сполучених Штатах, якому він очолював протягом 8 років. Це один з найбільших університетів приватних, і в них дуже цікаві, в тому числі медицинські дослідження, кому це буде цікаво, можуть подивитися. Крім того, сер Пол Нерс був 61-м президентом Королівського товариства. Це найвища установа в галузі науки в Великій Британії. І, звісно, сьогодні він донесе до вас цікаву лекцію на тему контролінг клітинного циклу. Відповідно, після цього в нас планується питання-відповіді щодо самої лекції, а потім в нас запланована 1-1,5 на спілкування з лауреатом вже на загальні теми. Тобто це буде мотиваційна бесіда, це вже не вперше ми практикуємо такий формат, він дійсно навіть краще працює, ніж звичайна лекція, де буде можливість запитати у вас, у лауреата, будь-які питання щодо життя, його життєвого шляху, наукового шляху, його мотивацій або якихось порад. Минулого разу запитували, як правильно публікуватися в гарних виданнях і так далі. Тобто сьогодні є унікальна можливість, і ця унікальна можливість створена Національним центром Мала академія наук України спільно з університетом Тараса Шевченка та громадською організацією Європейська молодіжна академія наук. Тому прошу до слова нашого гістя, Нобелівського лауреата, сера Пола Нерса. Будь ласка.
Well, good morning, everybody. I first have to apologize that I'm speaking in English, but honestly, you prefer me speaking in English rather than trying Ukrainian. I'm going to talk today about work from my laboratory, which I've worked on for many years, concerning how the reproduction of cells is controlled. The work is a little old-fashioned because I thought you might be most interested in hearing about the research in my lab that led to the Nobel Prize, and that was now quite a long time ago, nearly 20 years ago, that I got that prize. So you have to cast your mind back several decades, and the work that I describe is not how you would do this work today. I still have a very active research laboratory, and we could do everything I shall tell you about today in about 2 years rather than 10 years. But just forgive me for going back into history. I want to tell you why this is an important problem, and I'm going to go back a long way in history to describe that. And I'm going to show you how you can discover new explanations in biology relevant to medicine when you know very little about them when you actually start that work. I'm also hoping that you will get a sense of how science is done, a sense of the importance of teamwork and of collaboration. I also hope you will see that it can be quite an exciting adventure too, and I want to convey some of that excitement to you. So, some background, some history. Well, it starts a very long time ago. You see up there a picture from 350 years ago, more actually, and it was a picture drawn by Robert Hooke, who was one of the founders of the Royal Society. This is the oldest academy, science academy in the world. He was one of the founders in around 1660. And what he did is he used a microscope, and you'll see a picture of the microscope that he used up there. He took a razor, a sharp knife, and cut a very thin slice from a plant, looked at this under this microscope, and he saw all these little boxes that you see in that drawing that he carried out there. This was the first description of a cell. He called them cells because somehow it reminded him of a monastery where monks lived, and each of those cells were the rooms in which the monks were actually living. What Hooke did here was discover what we now know to be the structural basis of life. All living things are, of course, made of cells, as you know. But not only that, the cell is not only the structural basis of life, if you like, life's atom, it is also the basic functional unit of life. It is the simplest unit that has the properties of life. And this took some years to sort out. Up here you see a picture of a German biologist, a pathologist, Rudolf Virchow, and he made two very important conclusions, not only him, of course. The first is that every animal appears as a sum of vital units, each of which bears in itself the complete characteristics of life. That is, the functional basis of life is in the cell. He also said all cells from cells. That is, omnis cellula acellular in Latin. That is, cells arose by division of pre-existing cells. And this is a very important conclusion for what I'm going to talk about. Because here you will see a dividing human fibroblast cell. And since I have begun this talk, each one of you in your bodies has seen millions of divisions occurring just like this, even in the minute or two that I have been speaking. Why is this important? Well, there's three reasons. They're shown up here. The first is that cell reproduction, that is the division of a cell from 1 to 2, is the basis of growth and reproduction in all living things. So it's fundamental for all growth and reproduction and development. Secondly, it links the copying of hereditary material, that's the DNA, the copying of that into biology. It occurs through the cell reproduction process. And thirdly, it has medical implications because it's important for cancer. I'm going to very briefly describe those things a little bit more detail, just to put this in perspective. This is a mouse embryo that you can see here, and on the left, you will see where it came from. Every cell in every organism starts as a single cell and undergoes repeated division, reproduction, until it forms a ball of cells that then differentiate into the embryo. So this is why this cell division process is so critical for all growth and development. Do you find this exciting? I'm going to show you a picture here. Every one of you in this room used to look like this. This is a mammalian egg being fertilized by sperm, and every one of us was once a single cell, and every one of us has been formed by repeated divisions of this single cell. So it's important for the growth and development. What about the linkage with heredity, with DNA replication? What you see up here is a diagram of what I study. This is called the cell cycle. The cell cycle is the process by which cells reproduce themselves, and crucial and critical for every cell cycle are two events. The first one, labeled S at the top of this circle, is DNA synthesis. It's when the chromosomes, represented here by a blue line, it's when the chromosomes replicate. This is the double helix replication of Watson and Crick that you will be very familiar with. I run an institute in London named after Francis Crick, and Jim Watson came to open it, in fact. Not to open it, it was opened by the Queen, to be more precise, but he was present at the opening. So this is the copying of DNA, the hereditary material. But that is a chemical process. For it to be relevant to biology, it has to be translated into the biological sphere, and that's what the cell cycle does. Because after the chromosomes are replicated, they have to be segregated equally to the two newly dividing cells. That occurs during M phase, at the bottom of that circle, when each newly dividing cell receives a full complement of the DNA. So that is the second important feature, the linkage of the chemical replication of DNA into the biological sphere of cells that occurs every cell cycle as cells reproduce. The third aspect I want to bring to your attention is to do with cancer. I drew this little diagram up here to illustrate how cancer arises. You see all the blue boxes. These are behaving cells that are part of, for example, a tissue in your body. And they are under control. Their division is controlled very tightly. But if damage occurs, particularly in the DNA, that damages genes important for the reproductive process, and you see that's reflected there in a red box, a red cell that's gone wrong, then that cell can divide out of control. And it can form a series of red boxes there, which are the basis of a tumor. And that tumor, if it interferes with a tissue or an organ which is vital for life, causes cancer and causes death. So understanding cancer needs understanding of the cell cycle, understanding of reproduction of a cell, and how, in particular, it can be controlled. And that is the problem that my laboratory has worked on actually all of my life. So cell reproduction is important, and I wanted to ask the question, how is that controlled? And when this work started, we did not know any idea how it was controlled. And secondly, how can you investigate a complex biological problem such as this? I just show you a building. This is an institute that was in central London where I did a lot of this work. Actually, I worked there about half my life. It's just been demolished. It's no longer there. It's called the Imperial Cancer Research Fund, but the researchers there, I've moved into a new institute, the Francis Crick Institute in central London. This is a I won't talk about the Francis Crick Institute now, maybe later in questions, but it's the biggest biomedical research institute in Europe. It has around 1,200, 1,300 researchers under one single roof. And I'm the director of it. Okay, so the problem, as I said, is how is the cell cycle controlled? How can it be investigated? Well, a good way of investigating unknown problems in biology is to use genetics. Now, why is that useful? Well, the good thing about genetics is it allows biological problems to be investigated when almost nothing is known about them. Complex living organisms have, such as ourselves, have about 25,000 genes. Some very simple organisms have more genes than humans. Actually, small plants can have 50,000 genes, even more in some insects and so on. But 25,000 genes is quite a lot. Now, these genes encode proteins, proteins that carry out all the processes of life. Now, I decided to study this problem in a very simple organism. I'm going to show you a picture of it in a moment, which only has 5,000 genes. It's single-celled, and it's a yeast. And this single-celled organism doesn't have to make a body, it doesn't have to make a nose, eyes, hair, or anything of this sort. It just has to reproduce cells. So I used a simple organism to ask this question, so I don't get diverted by the complexity, for example, of our cells. So that's the first thing to be aware of. When you start a problem, in biology, choose the material carefully that you intend to study. Because I chose the simplest system so I could focus simply on this process of the control of reproduction. Now, how do you take on a problem? Like what controls reproduction? Well, using genetics is illustrated here. This box here on the left is the yeast I work on, fission yeast. As I said, I'll show you, well, I can show you a picture now. This is it, okay? So it's a cylinder, and this is a symbol of it, okay? And in there, I've got those green spots, and those green spots represent genes. There's 20 up there. There's 5,000 genes in this organism. Actually, my lab sequenced it. It was the third eukaryote to be sequenced, and we have all of the sequence of this organism, but not when I did this work, Now, what do? We're interested in cell reproduction, and what you can do is treat the yeast with a chemical or with a radiation that damages genes, and the genes, when they become damaged, which are involved in the process that you're interested in, can lead to what we call phenotype, an altered phenotype, which reflects the process that you're studying. I'm going to describe this in a little more detail here, but what you see at the top there in the middle are cells that are dividing. What you see beneath them are cells that can grow but cannot divide. So a simple approach, and I shall describe it again, is to mutagenize the cells, damage genes, and then select randomly for those strains which have got damaged genes in the process that you're interested in. So that's why you start knowing nothing, and you find genes that are important through this process. Let me just show you how that works. This is fission yeast. You can see it there with a scanning electron micrograph. This is the first picture of a fission yeast that I could find. It was drawn from a brewery in Copenhagen for Carlsberg Lager. The brewer when the fermentation didn't work and they made bad beer, they looked under the microscope and saw microorganisms that didn't do a good job, and they found fission yeast. So in fact, this is a bad thing for making good beer. So this is an illustration to show you what you have to get rid of if you want good beer. I think I'm telling you fission yeast is pretty useless, actually. Unlike budding yeast, which is more useful. Okay, this is a diagram. And by the way, what I'm showing you is all the diagrams I drew in the 1970s when I began this work. And so this is why it looks so old-fashioned. Now, I want you to look at this diagram. This is a representation of cells growing through the cell cycle versus time. The cell mass goes up. To the left, you see what I've called the normal cell cycle. A cell is born. You'll see the black blob is the nucleus containing the DNA. As the cell proceeds through the cycle, it grows, it divides, and it starts again. Now, I want you to do a thought experiment. I want you to ask if there's a set of genes amongst these 5,000 that are required for this reproductive process, and if they are defective, what would it look like? What would the cells look like? Well, many things can actually stop cell division. If you can't grow, if you can't make RNA, if you can't make protein, if you can't make lipid, if you can't make energy, all of these will stop cell reproduction. But they are irrelevant for understanding the process because they are just concerned with growth. So if you have a gene that is specifically required for the reproductive process, then what you will see is a cell that can continue to grow, but which cannot divide. Is that simple? You got that? And because fission yeast is a tube and it grows like this, the cells will get very elongated, as you see in the top right-hand corner. And this was the first screen I did in the 1970s to identify genes that were necessary for the cell reproductive process. Did 2 screens, took a couple of years, and identified, I think, about 25 CDC genes, standing for cell division cycle genes, right? And I'll show you a picture of what these cells look like. A is what a normal cell looks like. B and C are typical of the cells that are CDC mutants. This man over here is Kim Naismith, was my graduate student when I was working in Edinburgh. He's now professor of biochemistry in Oxford. So this process identified genes that were necessary for the cell cycle. Only identified some of them. I have to say now, we know that there's around 250 such genes that are required for cell reproduction. We know that because I told you we sequenced the whole genome of fission yeast, identified 5,000 genes, and then went through and deleted each gene one at a time, and then went back and looked for how many of those genes were required for the cell cycle, and that identified 250 essential genes. But we did that in the last 10 years. We didn't do it then. So at this stage, all we had was 10% of the total. So just a sampling we had at that particular stage. Now, this is a beginning, but you'll remember I was interested in what controls cell reproduction, what controls the cell cycle. So the question is, what do I mean by control? It's a term, actually, you need to think about. And in doing these genetic approaches, you have to be very careful about the way you use words, because words like control mean different things to different people. Now, what we identified with this screen was simply genes that encoded proteins that were necessary for the cell to complete the cell cycle. They didn't necessarily control that process. Now, there's 2 meanings for control I want to talk to you about today. The first one is what is rate limiting for the progression through the cycle? In other words, what determines the overall rate at which the reproductive process works? If I can use a metaphor, if you have a car and you want to know what controls the car, the speed of the car, what you can do is take pieces off the car and see what affects the speed of that car. If you take the door off, it doesn't affect the speed. You may get cold, it may not be practical, but you don't affect the speed. In fact, if you take the windscreen out, it doesn't affect the speed. The only way you can practically affect the speed is to start changing the amount of petrol that goes into the engine, and that's controlled by what we call the accelerator. So what you want to look for are mutants in the accelerator, using the metaphor of yeast. We want to affect the genes that control the speed at which the cell cycle can occur. Now, if you did, first of all, do such genes exist? They may not exist. But if they did exist, what would be the effect of them? What would it look like? And that's described on the next, which you've seen already, slide here. Look at the normal cell cycle again, and you see it takes a certain amount of time to complete the processes. If there's now a gene which is rate limiting for completing those processes, and you mutate that, then the cell cycle can be completed more rapidly than normal. If it's completed more rapidly than normal, the cells will divide before they've grown to the same extent, and they will simply look small. In fact, like these mutants here you see on the right. Okay? Now, I identified these mutants when I was working in Edinburgh in Scotland, and the Scottish word for small is wee. Have you heard of that? So I call these wee mutants. I thought it was funny in 1975, but telling the same joke for 50 years, it gets a little stale, has to be said. Now, the way I just described these experiments, it's as if I thought about this problem and then looked for wee mutants. That's how I described it, yes? Unfortunately, that's not true. I was simply looking for something else, those long cells under the microscope, and then I saw these small cells. And then I thought, what are these small cells doing? And then I went through the thinking that I just described to you in about 2 minutes, 1 minute, looking at them, and then realized that these mutants, which I'd identified completely by accident, actually revealed something about the control of the cell cycle. So that's something else about science. Science is often built on chance observations, on serendipity. You have to keep eyes open for things that you don't expect. And there is it's no exaggeration to say that the rest of the work I did was based on this chance observation. So, the wee mutants. This is the wee mutants. This was a colleague at the time, published 1975 in Nature, a journal you probably know. And it identified a gene that was rate limiting for cell cycle progress. Now, what can we do with that? Well, we could ask where in the cell cycle this gene acts. And that's possible because the very first mutant that I isolated was temperature sensitive. That means it worked well at low temperature, no problems, and it was only defective at high temperature. Okay? So this meant that I could take a population of cells, shift to the high temperature, and then simply ask the question, at what point do the cells start getting small? Because if it's at the beginning of the cycle, it will take a whole cycle before that happens. If it's towards the end, it will happen rather quickly. You see the thinking. Did that experiment and found that this gene acted at the onset of M phase, of mitosis, when the chromosomes separate. So this identified that this gene acted at the control of the onset of mitosis. So this simple experiment, and it's extremely simple, this is not the Large Hadron Collider in CERN, it's a very simple experiment, identified firstly that there were rate-limiting steps in the cell cycle, secondly, that they acted over the onset of mitosis, that is the end of the cycle. And thirdly, we had one gene that was involved in the process. The problem was, what could we do with that information? Well, knowing that these mutants reveal something means you could now go and look for more of these mutants. And so find how many genes behave like this. So I set myself the task of isolating 50 such mutants. But they're difficult to find. I could only find about 1 or 2 each week. So this took a year, or just under a year. Has to be said, this was very boring. Very boring indeed. And that's another aspect of science. Sometimes it is boring, and sometimes you have to do boring things to find out things that are interesting. Well, I did get 50 mutants in the end. And there's 2 things I wanted to say about them that were important. The first is this. I was looking for more than one gene. I thought maybe there'd be 5 genes or 10 genes that would behave like this. So every time I got a new mutant, I did a genetic cross to Wee1 to see if it was the same gene. And every time I did it, it was the same gene. And I went through 10, 20, 30, 40, and it was always the same gene. I began to wonder whether life was worth living after 8 or 9 months, because all I was doing was discovering the same gene. Then, one Friday afternoon in Edinburgh, in November, when it was cold and raining, I found a wee mutant, and it was covered on a plate by a fungus. This means it was very difficult to purify. I was tired, and I threw it away in the rubbish. I said, I can't work on this. I went home on my bicycle. I got wet. I had tea, and then I felt guilty. I felt I'd thrown away this plate. So I got back on my bicycle, 8 o'clock in the evening. I cycled back in the rain. I got it out of the rubbish. It hadn't been cleaned by them. And I started the microdissection, which took about a week to purify it from the fungus. This was the 47th or 48th mutant that I'd isolated. And you know what I'm going to say. It didn't define Wee1. It was a second gene, which I called Wee2. This is why I'm saying you think of nice names, but they get a bit stupid after a while. Not me too, by the way, Wee2. So this is Wee2. And this mutant was dominant. Wee1 was mostly recessive. And what does that mean in genetic terms? It means that Wee1 encoded an inhibitor, and Wee2 likely coded an activator. Now, if it was an activator, that is a gain of function which has more activity, what this suggests is that Wee1 was inhibiting the onset of mitosis, whereas Wee2 was an activator which had more activity. If that was true, then if you eliminated the activity of Wee2, what would it look like? It would look like a CDC mutant, because it couldn't divide. So I crossed Wee2 to the 25 CDC genes, remembering we only had 10% of the total. But we were lucky. Wee2 turned out to be a mutant of a gene called CDC2, which I'd already identified in the 25 genes. This was completely lucky. I only had 10% of the total, but one of the genes there was CDC2. So this told us that CDC2 was a key gene controlling the onset of mitosis. I found a photograph of myself when I did this. Thought I'd show you. I'm the one on the left, okay? You'll notice I had coloured hair then, and more of it then. This was a friend of mine who collaborated with me. This is a typical 1970s scene. Shall I leave it up there so you can laugh at me a little longer? Yes? Now, where are we? What we've done is discovered that there's rate-limiting controls. We've discovered that the controls act at the onset of mitosis. And we've discovered 2 genes involved in the process, WEE1 and CDC2. But then something else turned up, which was extremely important and was again accidental. And this goes back to another way of thinking about control. I told you that this was talking about rate-limiting controls. Another way of thinking about controls, which is related to being rate-limiting, is controls which actually switch on a process. In this case, the reproductive process. And the way this was approached in yeast was to think of it in the following way. Yeasts undergo repeated cell divisions. They can undergo sporulation. They produce spores. They can be pulled out of the cell cycle and undergo another developmental sequence. Yes? So what we could do is ask, where in the cell cycle do the cells become committed to the process of cell reproduction so they cannot undergo alternative pathways such as sporulation? You get the thinking? So what we could do in that experiment was to block cells at different stages in the cell cycle, challenge them to undergo sporulation, I won't talk about the technical details, and ask, can you sporulate or can you not? And what we expected was that cells early on in the cycle, before S phase, would not be committed, so they would be able to sporulate, and point later in the cycle, they would be committed. And that indeed was the case for nearly every gene. So what we found is that there were a couple of genes acting in what we call G1 before S phase, which could sporulate effectively before the start of the process. And all the genes, the other 15 or 20 that we looked at, are committed. And so those could not sporulate. The problem was, when I did the experiment with CDC2, remember CDC2 was the WEE2? I got an intermediate result. Instead of being nearly 0% or nearly 100%, it was 25% or 20%. And I kept doing the experiment again and again to get the right answer. The right answer was 0%, and I never got it. I got depressed, I put it away, I got it out again, I did the experiment again, I always got 20 or 25%. My difficulty was I didn't believe it, and so I didn't think about it. I wanted to get a particular result, and I wasn't getting it. I even thought maybe I should just forget it altogether and just publish everything else. I didn't do that, though. I didn't do that. Then, in the end, I thought, why don't I just imagine that the data may be true? It might be right. Could I explain it? And I came up with the idea, and it's shown here, that maybe CDC2 is required twice in the cycle. Required in the rate-limiting step for the onset of mitosis, but also required earlier in the cycle, before the start of the cycle, where it is important for committing the cells to DNA replication, S phase, and all the other events. And that when I took the mutant and shifted it to the high temperature, some cells were before start, although most cells were after. And that would explain the 20-25%. And it was true. What I could do is synchronize cells at the beginning, and they all conjugated. And if I synchronized them later, none of them conjugated. So this meant that CDC2 was very, very special, because it was required at the two major controlling points of the cell cycle. It was required at the beginning to start the cell cycle and to begin DNA replication, and it was required at the end of the cycle to finish it by controlling the onset of mitosis and then cell division. So it was the key master regulator of the cell cycle, at least in yeast. Now, you would have noticed, up until now, my analysis has been completely abstract. I've just talked about gene names and functions in processes, and we have no idea about the biochemistry or the molecules or what's happening here, because classical genetics does not allow you to do that. But the time now was 1980, and this was only a couple of years after the invention of DNA cloning. So I then spent 2 years working on DNA cloning, how to do it, and how to use fission yeast for molecular genetics. And the big problem there was, how do you introduce DNA into fission yeast? How do you make vectors that allow DNA molecules to replicate, and all the molecular genetic techniques that now, if there's biologists in the audience, you just take for granted. But you didn't take it for granted in 1980. That took me a couple of years. Now, why did I want to do that? Well, I wanted to do it because it allowed me to clone genes like CDC2. And that's illustrated the thinking of it here. Once again, you've got used to this now. Fission yeast is just the box. And in there, you'll see now a red line, which is the gene you're trying to clone. Let's say CDC2. Above, you see the gene library. Once again, I drew this in 1980. This is why it looks like this, okay? And what you see is the circles. And what I did is to make a gene library by cutting up fission yeast DNA and ligating it into plasmid molecules, transformed it into a fission yeast CDC2 temperature-sensitive mutant. These cells could not grow at a high temperature. But if I introduced into those cells a plasmid which contained the CDC2 gene, then it would rescue that defect. It would complement the defect. And this allowed you to select for the plasmid which contained the gene that was defective. This is the experiment we did. I isolated the vector with the CDC2 gene in it, and now these cells could form a colony on the plate. We could isolate the plasmid DNA from that and get the CDC2 gene. And that's how we cloned the CDC2 gene in the 1980s. Now, to find out what a gene does, remember we now want to know about molecular function. To find out what it does, you have to be able to sequence the gene. Again, there are some biologists here. Yes, a few, maybe? Okay. So this gene was 2 kilobases long. It had 2,000 nucleotides. It took me 1.25 years to sequence 2 kilobases. We can now do it in about 15 seconds. 15 seconds versus over 1 year. Can you imagine it? It took me over a year to sequence this gene in the laboratory. And I was collaborating with the inventor of DNA sequencing, Fred Sanger, to do it. Then when we looked at the sequence, did it tell us anything? Well, today, there are many, many, many thousands of gene sequences available. So you can just go onto the computer and you learn a great deal. At that time, there were about 50 sequences available. That's all, 50. And this gene, CDC2, from yeast, the only gene that it had any similarity to was a oncogene in a cancer virus called SARC-P60. And the only thing that was known about SARC-60 was that it was probably a protein kinase. They weren't sure, but they thought it was a protein kinase. What is a protein kinase? Well, a protein kinase is an enzyme, and it adds phosphates onto other proteins. Phosphates are bulky and negatively charged, so they disturb and alter the protein structure, and so change the characteristics of those target substrate proteins. So protein kinases can act as switches because they can affect many, many different substrates and change their function. I've illustrated that here. What we can see is the protein kinase on the right, phosphorylating a number of substrates, 5 here, as you can see. The kinase puts the red phosphate onto the protein changes its structure and changes its properties. In this case, it allows interaction with the green blob. So it changes, it can act very much as a switch. So I thought maybe CDC2 is a protein kinase, and maybe it's acting as a switch to switch on S phase and to switch on mitosis. But how do you prove that? Well, methods weren't worked out very well, but what I did was to take this CDC2 gene, put it into bacteria, express it in bacteria, make protein, put the protein into a rabbit, get the antibody from the rabbit, make an extract from yeast, and then use the antibody to purify the protein. Again, quite a few months. Now we do this all the time, of course, but again, this hadn't been done before. And I immune precipitated out of yeast, and I wanted to ask the question, does it have protein kinase activity? But the trouble is I didn't know what substrate to use. What protein could I put in there? So I simply went to a biochemical catalogue and bought 50 different proteins. And I put them all in the assay. And most of them didn't work. But one, a milk protein called casein, worked. I got some activity. The milk protein is irrelevant. It just happened to work in this assay. But I was so worried about this, is it just all spurious, that I did the following experiment, maybe shown on this slide here. Yes, on the second one. What I did is I made an extract from a temperature-sensitive CDC2 mutant and a wild-type cells. If you took wild-type cells, I immune precipitated this protein kinase activity, incubated it in vitro in a test tube at low temperature and high temperature, and it had activity at both. If I then did the same experiment with the temperature-sensitive mutant, the second one along there, and incubated it in vitro at low and high temperature, the kinase activity was destroyed at high temperature. This proved that the activity we were looking at came from that temperature-sensitive gene, and therefore the kinase assay was reflecting what the CDC2 protein is doing. This allowed us then to make synchronous cultures, and you see on the right here, what's a synchronous culture? We selected the small cells at the beginning of the cell cycle, grow them out, then we can sample throughout the cell cycle, measure the protein kinase activity, and ask how it changes through the cycle. And you can see, because it's the black circles there, that just before mitosis, the kinase is activated, and you get a peak of protein kinase activity. So now, through these methods, we could show that the CDC2 gene encoded a protein kinase, and the protein kinase is activated just before mitosis, and that brings about mitosis by building molecular genetics onto the genetics. This was done by my postdoc, Sergio Moreno, from Spain, and this is me again on the right, looking strange as usual. Well, we repeated this, cloning a variety of other genes, and we built up this network, and I want to just briefly describe it because it describes the control of the onset of mitosis. In the middle, you'll see P34CDC2. That's the protein kinase I just told you about. It forms a complex with another gene called CDC13. The protein there we now know is a protein called cyclin, and that activates the protein kinase. It's inhibited by WEE1. Remember WEE1? And that's at the bottom there. And WEE1 is also a protein kinase and phosphorylates CDC2 and inhibits it. We knew the residue, the amino acid residue in CDC2, it's a tyrosine 15, and that inhibits the protein kinase activity. CDC25, I haven't told you about that, is another gene which acts in this system, and that encodes a phosphatase that takes off the phosphate. So it's a balance between the WEE1 inhibitor and the CDC25 activator that controls when the protein kinase is activated, and we call these protein kinases cyclin-dependent kinases, CDKs, and that controls the onset of mitosis. Now, all of this was in yeast. Now, most people, I have to admit, are not interested in yeast. I happen to be interested in yeast, but most people in the world are not. And I found myself working in a cancer research institute who just thought I was a nuisance that would contaminate their tissue cultures with yeast, and I thought I have to do something to make them more interested in what I do. And I thought the best way to try and make them more interested was to see whether the same system also controlled the cell cycle in humans. But you have to admit there's quite a big difference between yeast and humans. Indeed, they probably separated in evolutionary terms 1.5 billion years ago, certainly at least 1 billion years ago, so it's a long time. So we set ourselves, and when I say we, I mean my postdoc Melanie Lee here, who worked with me, to isolate the, to try and identify if there was a CDC2 gene in humans. Well, we use conventional, remember the human genome hadn't been sequenced. Now we just look it up in a computer and do it in 1 minute, okay? It hadn't been sequenced. So how do we do that? Well, we could look for either proteins or DNA that were similar to the CDC2 gene or the protein in yeast. And there are methods to do that through antibodies and so on. We did them, and we isolated protein kinases from humans, but there's 500 different protein kinases in humans. So how do we know if we have the right one? So after a year or so of failure, I suggested that Melanie do this crazy experiment here. Now, you've seen this already, okay? It's the way we cloned the fission yeast gene, remember? But this time, we substituted the gene library from humans. So we put in it a human cDNA library. Actually, it was the first human cDNA library ever made. We didn't make it. It's made in Stanford in the United States by Paul Berg, and he sent it to us only 3 months after he'd made it, just as a gift, when I wrote to him. I put into the yeast cells this gene library, and amazing, looking to see whether there was a gene in human beings that could do the same function as the yeast CDC2 gene, right? So I was asking for the function to remain conserved for over 1 billion years. I mean, who thought this experiment would work? I mean, I didn't, but I thought we should try it. But it did. We pulled out a human clone, and the clone rescued the yeast mutant, suggesting it has the same function. We had to sequence it. This time it only took about a month to do it. Still took a month or two. And this was done by Melanie Lee. And we had an amazing result because the protein we got from humans was exactly the same size, only 1 amino acid different in size, over 300 amino acids, and it had 61% identity across the protein. And this is 1 billion years plus divergence. So the whole system was totally conserved from yeast to humans, and therefore in every other living eukaryote that you can actually see. So this showed that the control system that we'd worked out in yeast also applied to humans, and almost certainly, therefore, applied to every organism in between, so it was highly conserved. I took a crude photograph down the microscope of the first complementation experiment, which is here, the first experiment where a human gene is rescuing a yeast mutant, and it was on a plasmid vector, and when you lost the vector, the cells could no longer divide. And do you see those long elongated cells there? That was because they lost the vector. Well, I'm going to stop in a couple of minutes, okay? But I just want to summarize how we now know this is working. And I'm not going to explain the experiments by which we worked this out, but I can possibly do a bit more in questions. We now know, although most eukaryotes have a family of cyclin-dependent kinases, you can control the cell cycle with only one cyclin-dependent kinase. And the control is incredibly simple. I've drawn it out here. If you look to the left, at the beginning of every cell cycle, it starts with low activity. And to undergo S phase, you have to get a small amount of activity. Then, when you've completed S phase, you get more activity, and that inhibits a further round of DNA replication. So there's only one round of S phase in each cell cycle. And when you get more CDK activity, you get mitosis. So you order all the events of the cycle that matter through one enzyme, increasing activity as you go through the cell cycle. Couldn't be simpler. And then you destroy the cycling component, and it all goes down to a very low level, and the whole thing starts again. So the actual model is incredibly simple. It's not the way most people think about it, but it is the right way to think about it. Okay, this is a summary. So I've shown you that cyclin-dependent kinases control the cell cycle, that their increasing activity, which is regulated through the cell cycle, controls and drives the onset of DNA replication. It inhibits, which I haven't explained the experiments we use, when the activity gets higher, it inhibits S phase, and then it brings about the onset of mitosis. And furthermore, this control is conserved in all eukaryotes. I want to end with another quote. This is a quote from Theodor Schwann in 1839. Schwann and Schleiden was one of the proponents of the cell theory. Let me read it to you. 1839, in German, of course, translated into English. We have seen that all organisms are composed of essentially like parts, namely of cells. That's the cell theory. That these cells are formed and grow in accordance with essentially the same laws. Hence that these processes must everywhere result from the operation of the same forces. That was the speculation in 1839. We showed it was true in about year 2000, but it did take another 150 or 170 years. But we were anticipated by Theodor Schwann. Thank you very much for listening to me.